Since 2006, the John Paul II Medical Research Institute has established itself as a pioneer in advancing ethical adult stem cell research and regenerative medicine. Due to the gravity and urgency posed by the current COVID-19 pandemic, JP2MRI decided to shift a part of its research focus towards developing the most definitive vaccine platform for addressing COVID-19 and emerging coronaviruses. The primary reason for our decision stems from a critical deficiency in the current approaches being used to create a safe and effective vaccine. While the government has spent billions of dollars through Operation Warp Speed to try and develop several vaccine candidates against COVID-19, the companies developing these vaccines have chosen to use approaches that lack a historic track record of success. These less effective approaches involve targeting a fragment of the COVID-19 virus called the spike protein. Unfortunately, fragment-based vaccine approaches have failed to produce FDA-approved vaccines after the outbreak of SARS 2002. Additionally, a major challenge in producing a potent vaccine is the requirement of a human cell line which closely resembles what the vaccine is intended to treat. The current cell lines used in biomanufacturing are outdated and not as robust as needed to mount an effective response. Moreover, some of the current vaccines in clinical trials were created from aborted fetal cells. JP2MRI is using a historically proven, tried-and-true approach in developing an ethical vaccine, one which is considered to be the gold standard by leading virologists and which is being ignored by other vaccine developers in their rush to market. Let us highlight the advantages of our vaccine platform.
Historically, vaccines takes years to develop. First, vaccines have to be tested in animals for safety and efficacy before they can be tested in patients in clinical trials. But in the case of Operation Warp Speed, some of these vaccines have gone from design to manufacturing to clinical testing. And what's concerning is that many of these vaccine methods have not had a track record of success and have never produced an FDA-approved vaccine before. And some of these vaccines are using a virus called adenovirus to carry the spike protein into patients. And these adenovirus-based vaccines are manufactured from aborted fetal cells. What is particularly important to note is that these adenovirus-based vaccines were historically used in the past to create vaccines against HIV. What actually happened years ago was that these particular vaccines actually increased the risk of HIV. And so those vaccine clinical trials were terminated. And lastly, there's a concern that delivering a spike protein of the COVID-19 protein may actually increase the toxicity of those vaccines, because there's a segment of the COVID-19 protein that has some similarity with segments of bacteria and viral proteins that are known to cause toxic shock. So we have to be very careful about designing public health policy on the basis of these Operation Warp Speed vaccines. And we have to have some realistic expectations for the public about the outcomes of these vaccine programs.
Many leading virologists around the country have raised concerns that the U.S. is applying too much of its financial resources towards vaccine development methods that have a very poor track record of prior success, while ignoring more established approaches using inactivated or killed virus vaccines, such as those used to create the flu vaccine, and live attenuated virus vaccines, such as those used to prevent polio and smallpox. Inactivated and attenuated live virus vaccines have been shown to provide much more long-term immunity, since they elicit cell-mediated immunity by providing a greater number of viral targets. However, attenuated live vaccines take more time to develop than fragment-based vaccines. Should the rapid U.S. vaccine approaches fail, and should other countries like China that are working to create a live attenuated virus vaccine succeed, it will have far-reaching implications to our country. The Institute is pleased to report that by working at record pace and under budget, we have already developed a novel method of producing immortalized human cell lines for vaccine development. We believe that our cell lines will be much more effective in producing a successful vaccine. JP2MRI's technology therefore offers not only an ethical solution, but also one with significant scientific advantages. The Institute is now working on the first phase of our research platform to produce a prototypic attenuated live vaccine. It is anticipated that the cost for Phase 1 will require $325,000 and will take 6 to 9 months to develop if sufficient funds can be raised. Phase 2 will require $1.5 to $2 million and an additional 1 year to complete animal testing to evaluate safety and efficacy before clinical testing can begin. While this timeline is longer than the best estimates of Operation Warp Speed, the outcome of our research represents an important backup strategy for COVID-19 and, more importantly, a long-term solution to address any future novel coronavirus outbreaks. The timelines noted will be contingent upon successful fundraising by the Institute. Your financial support will play a critical role in helping JP2MRI develop an ethical and effective vaccine platform for addressing COVID-19 and future coronaviruses, particularly if COVID-19 mutates and becomes resistant to current antiviral treatments. To donate and to learn more about our approach, please visit jp2mri.org. We welcome you to also follow our research progress by joining us on Facebook, Twitter, and YouTube. Thank you for your support.